of experience with vaccine development across all stages of both the clinical and preclinical research.
Thank you, Thea, for the kind introduction. I'm happy to introduce our ECCMID appointed chair, Thea Carlson-Fischer, to you. Thea is joining us from Copenhagen, where she's a professor of public health at the University of Copenhagen, background in MD and trained in infectious diseases and virology. And thank you, Thea, for being our co-chair.
Thank you very much. My pleasure. And also, the stars of today's shows are not the two of us, but the two presenters. And I'm pleased to introduce Professor Adam Finn, whom many of you probably already know quite well. Adam Finn is a professor of pediatrics at the University of Bristol and the head of the Bristol Vaccine Center. He has a long standing career within both WHO and within the Health Joint Committee and Vaccination and Immunization Program at the British Department, and has also formerly been the president of the European Society for Pediatric Infectious Diseases. Welcome, Adam.
Thank you.
Great. Next to Adam and today's other speaker is Professor Thomas Jelinek. Welcome to the session, Thomas. And Professor Thomas Jelinek is the medical director of the Berlin Center for Travel and Tropical Medicine, as well as the scientific director of the Center of Travel Medicine in Düsseldorf in Germany. We very much look forward to both the interviews and the following sessions, presentations provided by you. Thanks, and hereby the floor is yours, Catherine, for starting off the interview.
Thank you, Thea. So we're starting this off with an interview with Adam Finn today. So Adam, you've been involved in policy development for the UK COVID-19 vaccine program as a member of the JCVI. Can you tell us about the early stages of the formulation of the plans that were put into effect late last year?
Yes, thank you for that question. I think one of the really key parts to the success so far of the UK program is the planning that took place during 2020 before we actually knew whether we would or when we would receive vaccines. And a lot of work was done on the epidemiology of the COVID pandemic that we were experiencing in the UK during those months, so that the JCVI started meeting on a regular basis as early summer 2020 to discuss this, and in particular to identify the risk groups who were most likely to be affected by the disease. Obviously, elderly healthcare professionals, elderly folks, particularly those living in residential accommodation, and then to identify the comorbidities that predicted severe disease, so that we were already well advanced when it came to the end of the year and we began to have vaccines to use with a strategy that was already pretty well formulated.
I see. And then finally, when the vaccines were available in January, the JCVI recommended a longer dosing interval for the two vaccines that were available then. How did you come to that decision? And in hindsight, was it the right call?
Yeah, well, this caused a lot of consternation at the time, actually. As we began to receive the data from the Oxford AstraZeneca vaccine, it became apparent that there had been different dose intervals used, almost by accident, actually, in that program. There was some confusion around the responses initially relating to dose, but it became clear quite quickly that the longer dose interval, around 12 weeks, resulted in quite substantially greater post-dose 2 responses. In fact, the response after one dose to that vaccine in terms of neutralizing antibodies were pretty good. So on the basis of epidemiological models, we were able to predict that many more deaths would be prevented by using the limited supplies to give first doses during that first 3-month period than to give second doses. The controversial bit was extrapolating that to the Pfizer vaccine, where there were no such data. All there was a small amount of evidence that in the interlude, the 3 weeks, or in fact really the 1 week between 2 weeks after the first dose and the administration of the second dose at 3 weeks, there was already apparently a protective effect being seen, even though the immunogenicity was not great. So that was done a little bit on a leap of faith. But in answer to your second part, well, it has definitely turned out to have been the right call, because we are seeing good protection after the first dose, much enhanced protection after the second dose. It's enabled us to get more people immunized during the early phase of the epidemic this year. And now we're seeing the benefit of bigger post-dose 2 responses as well.
Yeah. Okay. That will be probably helpful when it comes to the variants particularly. Now, you spoke about the uptake of the vaccine, and in general, I think the coverage rates that are reached in the UK program are very high. Although the plateau, and particularly looking at younger cohorts, they seem to plateau off at progressively lower levels. So what was the success factor in getting high acceptance rates in general, and what do you think could be done to get even better access in younger people who aren't so concerned with the infection?
Well, we have one great benefit in the UK to our advantage, which is that there's vast public trust in the National Health Service. This is a kind of love affair between the UK population and the health service. And so things that are delivered through the NHS are by and large very well trusted. I don't think we trust our government any more than anyone else does, but we do trust our health service. The other thing is that the sort of central planning that we have means that we deliver vaccines very efficiently and very close to home, usually in the primary care sector. And again, that makes access really easy for people. So that combination of trust and easy access, I think, has contributed a lot. Of course, fear of the disease is a big part of this, and that's why we've really achieved extraordinarily high coverage in the elderly. But the young are more of a challenge, and in particular now that we're getting a political message that things can be relaxed, I've got real concerns that young people will no longer see this as a priority. Nevertheless, we're now also beginning to see restrictions lifted for people who've been vaccinated, and that may provide some kind of incentive as well.
Yeah, I think this is also a broad scheme in many countries, the public lifting of measures. The vaccines that are used to prevent SARS-CoV-2 are very diverse at the moment. How do you think about that? Do you consider this a good thing? Do you think there will be even more diverse vaccines evolving in future, or do you think there will be a focusing down on only maybe 1 or 2 preferred vaccine platforms?
I'm strongly in favor of there being diverse platforms. I think we have already seen that although the novel platforms that have delivered the vaccines most quickly, that they're not platforms we're very used to using, and already we are beginning to encounter unexpected, rare, but serious problems with them. Also, we have to think about the capacity to manufacture vaccines and potentially revised vaccines in the future as the virus evolves. And so for all of those reasons, I think we really should do everything we can to have as wide a range of tools as we can get as we go forward in the pandemic. We will find that these different vaccines will also be able to use together, potentially successfully, so that people can receive different vaccines for variants, for boosters, and so on. So diversity, I'm sure, is the way forward.
Mm-hmm. I see. And when it comes to comparing the vaccination program against COVID-19 to other vaccines, what do you see as the relative importance of direct protection versus indirect protection? Is it similar than for other programs? Is it different?
This is a topic I'm very interested in. I spend most of my time researching indirect effects of vaccines. Because of the high efficacy against serious disease that we've consistently seen so far with the available vaccines, the programs that have been recommended by WHO and national governments have been very focused on direct protection. So elderly people who probably don't transmit the virus very much, nevertheless, the primary targets. However, as we go forward, the importance of indirect effects is just going to grow and grow. And we really need to know more about this and understand better how well we can bring about those effects. They are, of course, really important in just about all of the other programs of vaccination that we use very widely, with the possible exception of tetanus, where we acquire the infection from the soil and not each other. But indirect effects of vaccines are clearly vital. And at this critical stage in the pandemic, as at least in some countries we begin to get reasonable coverage, they're going to be bigger and bigger part of the success.
Yeah, so it will be truly interesting to see how that evolves as the vaccination rates grow. Absolutely. So coming to my last question, Adam, what do you think we need to do to accelerate progress in combating the pandemic with vaccines? What more do you think can be done or should be done?
Well, that question is one that could provoke a very long answer, but to keep it short, I think we've got to improve our global thinking. I think there's a little bit too much national thinking at the moment. And of course, the privileged countries that have the resources to buy the vaccines are taking care of their own populations. But if we don't think globally, we're going to be in trouble because, as we've already seen recently, we will see evolution of viral variants in areas where there's high transmission going on, and those will inevitably be transported around the world. So far, the evolution has been towards more transmissible viruses, which are very problematic. But the next phase will be viruses that are evolving to evade the vaccine-induced immunity to some extent that we've created with the vaccines we've got. So we need to think globally. We need to think how we use the vaccines that we have available over the next 12 months most cleverly in order to get the most control of viral replication, or we're just going to go on and on having problems.
I see, and I agree with that notion. So I'm just quickly checking to see if we have any questions from the audience being posted at the moment for Adam, but I think not yet. So I think we have a slight light interactive question for our audience to participate in. So
there's a question in the panel as well.
Yes, I've seen it popping up. So maybe we can take that question on. Adam, what are your thoughts concerning the vaccination of children, 12 or adolescents, 12 to 17 years?
Okay, so this is a tricky question for me to answer because the JCVI has actually issued recommendations on this to the UK government, who are deliberating on what to do, and we've been given very strict instructions not to speak publicly about it until an announcement is made, in case it's interpreted as being JCVI policy. But I will nevertheless try and answer the question. The truth is that this is not a straightforward question to answer. I think the easy part is that we do need to identify those children who are at similarly high risk as the adult groups to whom we've targeted these vaccines, because there's absolutely no reason at all why children should not benefit in the same way as adults for direct protection. But that is the minority of children, and we've seen this week data published from the UK that have given a lot of detail actually on what we've experienced in terms of pediatric disease. The two other factors that are more unknown really are the so-called long COVID or post-COVID syndrome, which for the moment does appear to be less of a problem in children than in adults, but nevertheless is a problem and can't be ignored. But perhaps most important of all is the disruption to education, which has been the biggest harm that children have experienced across our countries during this pandemic. So all children have had massive disruption to their education. The question, though, is whether vaccination is really a solution to that problem, or whether we should instead consider more carefully how much we really do need to disrupt their education going forward, because I think the evidence suggests that the amount of transmission and generation of the epidemic in schools has been really very modest. The data we've seen suggests that children's infections reflect what's going on in the local community around them. And in contrast, for example, to influenza, where we see epidemics driven by transmission, particularly in young children, we've not really seen much evidence of that for COVID. I think it might be more straightforward if we had a superabundance of supply of vaccines and we could immunize everyone, which we don't have, globally at least. And it would be easier if we had complete confidence in the safety of the vaccines we propose to give. Whereas for the only vaccine that's currently licensed below the age of 18, there is a signal suggesting that males, particularly after the second dose, may experience myocarditis, albeit mildly, but with no data so far on long-term follow-up. So you can see that there are a number of competing issues here, which make it really quite difficult to decide what would be best with regard to pediatric immunization. But I go back to my answer of your previous question, which is that we do need to think globally. And if we've got limited supplies of vaccines at the moment in the world, they probably should be being given to the people who are most likely to get sick, and that isn't children.
Great. Thank you very much. That was good questions and interesting answers. And I afterwards would like to follow up also, Adam, and hear a little more about your mentioning of the indirect outcomes of vaccination, and how about the non-specific outcomes of vaccinating, in particular children. I'm very curious to hear about that, but it will have to be at another time, because now it's time for you, Thomas, to take in the scene together with Catherine, and we'll hear some more interesting interview session from the two of you. So thank you very much.
Thank you, Thea. So, Thomas, the COVID-19 pandemic has been, of course, a real challenge for the travel health sector. So what were the strategies in your travel clinic setting to overcome the obstacles? How did you deal with that phase?
Yeah, thanks, Catherine. Obviously, travel decreased in an amount nobody could imagine before, and therefore counseling of travelers decreased as well. So we had to cut clinic times, even close some of the clinics temporarily, and deal with the situation. Strangely, though, the requests for information increased because, of course, people were very insecure about the effects of COVID-19 on an international basis. And we still had travelers, of course, who had to go for business reasons, humanitarian reasons, and others. So we did a lot of counseling, which was much more intense than before, and which way more question marks, really, because we didn't have the information either in many instances. So the workload per traveler increased actually significantly. And then, of course, new jobs popped up like PCR testing, which you need for travel. And you have to do that quite fast because there are time limits for travelers, really, when the PCR result has to be produced, certifications, and now, of course, there's COVID-19 vaccination, which we do offer as well. And now we see an increase again in traveling, tourism too, a slight increase. It's sort of waking up. And so we opened our clinics and are starting again, but we do see a higher effort, really, which we have to put in for everybody. Incidentally, if talking about easy access to the COVID-19 vaccines for young people, travel clinics are actually very well set to have easy access for them, because that's where they come when they want to travel, and then we can offer the vaccine easily. So it's been very well received now that they can get the vaccination against COVID-19 too at our settings.
Mm-hmm. Mm-hmm. I see. And you mentioned you still had some travelers that needed to take more time, business travelers, for example. Did you also see, for example, an increased interest in other vaccines? Did you see more uptake of standard immunizations during that phase?
On the contrary, there was a time when and there's still a time where we still actually have to fight to get the other issues through. Everybody wants to talk about COVID-19, and there's a decreased interest in other diseases and other vaccinations. It's just so much space is taken up by this one diagnosis, and we really struggle to make clear that yes, there is COVID-19, of course, but there are also quite a few other issues that are still there, like dengue fever, which is a pandemic since many years ongoing, and we tend to ignore it really on an international basis. Like malaria, like many vaccine-preventable diseases. So there are many issues that we try to get back on the agenda with the travelers, and that's a bit of work really in the moment.
And do you think that this impact of COVID-19 on travel medicine, do you think there will be a long-lasting impact, or an impact that remains? Once travel has maybe gotten back, do you think that will change people's attitudes towards vaccination in general by increased awareness?
Well, in the moment, of course, there's a lot. There's an impact, and will be felt for a few years. COVID-19 will be around, and it will be an issue, but it will become more and more just one of the many vaccine-preventable diseases we have. It will the situation will normalize, and then I do think that for a time, awareness towards vaccination will be increased, the acceptance of vaccination raised by those people that visit us, because becoming vaccinated is more like a normal process right now. And of course, the issue of certificates needed, PCR tests needed, will keep travelers busy. Right now, we really have an issue with or problems with vaccinated travelers, because there's a huge difference in countries accepting vaccines. So like US are not accepting some European vaccines, China is not accepting European, we don't accept Chinese vaccines, and then vaccination schedules are not accepted even within Europe. So it makes a lot of work, and that needs to normalize. In the long run, I think people tend to forget. The more normal COVID-19 will become, the more the awareness of prophylaxis will decrease again.
I see. So, do you foresee in the long run that the safety measures that we're seeing in place now for travelers, do you think that will stay in the long run?
It's our job to work on that, really. As I said, I think travel will normalize over the next 2 to 4 years back to quite normal levels. That's what we see right now. There are many people actually waiting to travel again, and quite a few just have to for their job or for other reasons. And so for the moment, awareness will be increased, and as I say, it's our job to keep it that way. But the more normal COVID-19 becomes, and the more people will be there that are vaccinated, the more people will forget that there was an issue. Really, we have seen that in the past too, with big pandemics. People tend to forget.
Yeah, that danger certainly exists. So I think we have time for a couple of questions. Thank you, Thomas, for the interview. I'm just checking to see whether we have any questions at the moment. Otherwise, I would have. Here we go. No, otherwise, we have a poll question coming up. So on a scale from 1 to 10, how much did you miss traveling yourself? And I think we should be able to see the results to the polls towards the end of our session.
Yeah, we will have to stick up on for the very end to get those exciting results. So very good cliffhanger here.
Yeah.
But
so
more exciting would be the next minutes, right? 30 minutes of presentations.
Correct. So I think we can go ahead and Adam, do you want to continue?
Sure, I will share my screen, and hopefully we can get the PowerPoint slide up. Could you just confirm you're seeing my title slide? Okay, great. Thank you. So thanks now again for the invitation to this afternoon. As you can see, my title today is Leveraging Established Technologies and Inactivated Vaccine Candidate, VLA 2001, with a novel adjuvant combination to protect against COVID-19. So my name, as you know, is Adam Finn. I'm a pediatrician from Bristol in the UK. At Adam H. Finn is my Twitter handle. If you're a Twitter person and interested in vaccines, please feel free to follow me. Just quickly, in terms of my personal interests, the hospital at which I work has received funding for the study I'm going to tell you about. None of that funding came to me. And the university that employ me has also received funding for the preparation and delivery of this talk. Again, that money isn't paid to me. I do and lead research funded by a number of different vaccine companies, both COVID-19 vaccines, but also many other vaccines in children and in adults. And finally, I'm a member of the UK National Immunisation Technical Advisory Group, the Joint Committee on Vaccination and Immunisation, and I chair the World Health Organization European Technical Advisory Group of Experts on Immunisation. I don't own any intellectual property, stocks, shares, benefits in any pharma companies or in fact in any other companies either. The vaccine I'm going to talk to you about over the next 10 minutes or so is a beta-propiolactone inactivated whole virion SARS-CoV-2 vaccine. You'll know that beta-propiolactone is an alkylating agent that's widely used because of its ability to disrupt both DNA and RNA and to leave the virus antigenically relatively intact. So it's widely used to inactivate viruses in the lab and for vaccines. The adjuvants in this vaccine are aluminium hydroxide, and most of you will know that aluminium salts, hydroxide and phosphate are used in just about all inactivated vaccines and have been for many decades. But also a cytosine phosphoguanine oligodeoxynucleotide, which is a novel adjuvant. This is a motif, a nucleic acid motif that's common in bacterial DNA. And these oligodeoxynucleotides bind to toll-like receptor 9 in innate immune cells and stimulate the immune response. So this is the novel part of the adjuvant of this vaccine. The study I'm going to tell you about was initiated at the very end of last year, around Christmas time. It's a phase 1/2 first-in-human dose-finding and safety study. And the plan was to enroll 150 healthy adult volunteers aged 18 to 55 years, and to give them all 2 doses of the vaccine 3 weeks apart, administered intramuscularly into the deltoid muscle. The 150 subjects were to be randomized to 3 different doses of the vaccine, and there were no controls who were given placebo or no vaccine. We did this fairly cautiously with 5 sentinel subjects, open label, initially to the low dose with 1 subject and then 2 and 2 over several days, and then dose ascending up to 15 sentinels over the 3 groups. And once that all went fine and there were no problems, we then randomized the remaining 45 per group to the 3 different doses in January. The study was conducted at 4 UK sites, which I'll give you the details of at the end. The follow-up is actually ongoing to this study, although the subjects are just reaching the time of their final visits at this current month, and will be offered to enrollment into a further booster phase of the study. So the study went well. We recruited to time and target, in fact, slightly over target. 153 people were enrolled, 51 per group within the enrollment period. And of those 153, 150 completed to the point of the data cut that I'm presenting, with 3 people dropping out. 1 got pregnant, 1 developed self-limiting but unexplained hematuria, and 1 got COVID. We have had more recently a few people dropping out as the national program progressed and people wanted to be unblinded and receive routine vaccine. The median age of the participants was 31 years, similar across the three groups. There were slightly more males than females, and it was a predominantly white group of people who were enrolled. And although I don't have time to give you the full details of the demographics, the characteristics randomized across the three groups were very similar. So onto some data. This slide shows you solicited adverse events after the first vaccination dose. What you're seeing here are triplets. Blue is the low dose, the pink is the medium dose, and the orange is the high dose. And you're seeing local reactions on the left side of the graph and systemic symptoms on the right side of the graph. What you can see here is that there's little evidence of a dose effect, so we're not seeing more side effects in the higher dose groups in any statistically significant way. And although we don't have any control group who received placebo or no vaccine, the kind of rates for these non-specific symptoms is somewhat similar to what we see when we do control studies of that kind. These are the same data for the second vaccine dose, so no real change here. The y-axis is almost the same for those two graphs. No dose effect seen, and no real signs of serious problems with local or systemic reactions. In terms of unsolicited adverse events, there were 60 recorded, just under 40% by day 36. One was considered serious. I'll tell you about that in a moment. 27 were considered treatment-related. These were largely raised erythrocyte sedimentation rates, biochemical abnormalities on blood tests, or odd people who developed dizziness or muscle pains. And only 4 of these were medically attended. There was no dose effect observed. In other words, we didn't see more adverse events in the higher dose group than in the lower dose group. The one serious adverse event, which was not considered treatment-related, was this adverse event of special interest that one subject developed chilblains, which is the swelling and redness of the toes 4 days after dose 1. This is of interest because it is a feature described in COVID infection. But in fact, this subject didn't have a positive PCR, received the second dose without any further effects. So likely they got their cold, itchy toes because it was wintertime. There were 2 COVID cases, one shortly after the first and one shortly after the second dose, which were mild and moderate in severity. We used 3 different immunological methods to evaluate the immune responses to this vaccine in this study. The first was a microneutralization assay with an ND50 rather than ND90 endpoint. This is an assay that's run by Public Health England in their Porton Down labs. It's the same assay being used for the AstraZeneca studies and in fact all the studies that are being sponsored by the UK Vaccine Task Force. It involves infecting Vero cells, which are African green monkey kidney epithelial cells, with the virus pre-treated with heat inactivated serum. And then the plaques, when the virus infects the cells, are visualized with an anti-spike protein receptor binding domain antibody. There was also a more straightforward binding assay, immunoassay, using a pre-fusion spike IgG approach, so with an IgG detection antibody and binding to the spike protein on the plate. And then finally, to measure cell-mediated immune responses, an interferon gamma ELISpot run by a lab called Oxford Immunotech, which again have been doing all the assays for the UK Vaccine Task Force studies, was used. This employs both nuclear protein spike and membrane peptide pools from the virus and has a 6-spot minimum cutoff to report detection of a response. So this assay also being used for the other vaccines being trialed in the UK. So here are the results from the neutralizing antibody. You're seeing day 1, 8, 22, and 36 across the graph. So pre-vaccine, 1 week post on the day of the second dose, and then day 36 after the second dose. And then these triplets are the 3 different dose groups, low, medium, and high in each case. And I think what you can see clearly from this graph is a dose-dependent rise that is clearly seen after the second dose of the vaccine. To give you some frame of reference, these titers are similar, in fact, somewhat higher than the seen after 2 doses of AstraZeneca using this assay in the same lab, and higher than is generally seen in studies using this assay with convalescent sera. So these were encouraging results. Just looking at the day 36, same data, but in a different way. This is reverse cumulative frequency on a log scale. So you're seeing in blue the low dose again, in pink the medium dose, and in higher on the right in orange. And you can see the distribution of the titers going across as we increase the dose. So a clear dose-dependent increase in neutralizing antibodies. I'm only going to show you one slide of the immunoassay, the binding antibody assays, and this correlates the immunoassay results on the y-axis with the neutralizing titers that you've already seen on the x-axis, and shows a close correlation between the two, which indicates, I think, that the immunoassay may be quite useful going forward as a much more straightforward way of measuring immune responses, which correlates well to neutralizing antibody. But as you will all know, we do not yet have a clear serological correlative protection for any of these vaccines. Finally, cell-mediated immune response. I'm just going to show you one slide, which is with the spike protein peptide pool. There are similar results for the membrane protein and the nuclear protein peptide pools. You're seeing day 36 for the low, medium, and high dose on the right-hand side of this graph, and the pre-vaccine responses on the left side at day 1. So perhaps gratifyingly, really, in the context of an inactivated viral vaccine, we are seeing evidence of a robust cell-mediated immune response here, perhaps attributable to the adjuvant that's being used. This is clear at the medium dose, and not really noticeably increased at the high dose, but clearly higher for the medium and high dose than it is for the low dose recipients. So in summary, the safety profile of this vaccine, VLA 2001, was within the expected range for an inactivated adjuvanted vaccine, and there were no safety signals detected in this small study, certainly none in any way to hold us back from further development. The high dose group has been selected for fairly obvious reasons for the phase 3 development stage, which is now ongoing, and in fact fully recruited and in follow-up. That study will be a superiority comparison with the AstraZeneca vaccine, and the safety profile and the immunogenicity results support that way forward. I'd just like to finish by thanking many people who've contributed to this study, most in particular the volunteers who took part, as well as the medical nursing and admin teams at the 4 sites. Valneva for sponsoring the study and providing a lot of support, and the UK government and the Vaccine Task Force, again, for both funding and practical support. On the slide, you can see the 4 principal investigators from the 4 sites: Chris Duncan from Newcastle, Chris Green from Birmingham, Saul Faust from Southampton, and Rajeka Lazarus from Bristol. Thank you.
Thank you very much, Adam, and thanks for keeping the time just perfectly. So now we can open the floor and take questions.
Okay, can you reassure me that the screen share has stopped?
It has stopped, and
yep. Right.
The chairs and the presenters.
Fine.
So thank you, Adam. I don't see we have any questions raised at the moment, so maybe in the meantime, as we wait for people to sort through their minds and type in their questions, Christoph, do you want to bring up a poll?
And I think while everybody is doing their best to guess, not hope, but guess, I would like to raise one of the questions from the audience, Adam, for you. First of all, thanks for your presentation, and then a question about what are the ELISpot responses comparable to the other vaccines?
Sure. Yeah, we can make a direct comparison actually with the AstraZeneca studies because it was the precise same assay. And actually, the AstraZeneca vaccine is extremely efficient at inducing cell-mediated immune responses, particularly after the second dose. So we can say that they're comparable. But in fact, I think the adenoviral vector vaccines are likely to be superior. Nevertheless, for an inactivated viral vaccine, the fact that we're seeing cell-mediated immune responses at all is actually very encouraging because that's not something that's widely expected to be present or indeed reported from other simply alum-adjuvanted whole virion vaccines. We don't, of course, know how much of a cell-mediated immune response you need to be protected, but the evidence is certainly emerging that this is an important part of the protective response, particularly perhaps when it comes to prevention of severe disease. Nevertheless, I'm not saying that neutralizing antibody doesn't matter because there's good evidence for that too.
Thank you. And there's a new poll.
It's the same one, isn't it?
But should we then be able to see the results?
So if I'm not mistaken, our audience should be able to see the results. It's just that as speakers, we are left in the dark as to how our audience actually replied. So
it's going to go on forever.
But I think we have another question from the participants. So the question is, how comparable will the Valneva vaccine data be to other inactivated vaccines? An example, could it be used to guesstimate how other inactivated vaccines, how well they may be doing?
Well, I'm not sure that we can draw a comparison very straightforwardly from these immunogenicity results, simply because we don't have other immunogenicity data from the exact same lab, certainly not the neutralizing antibodies. On the other hand, we do have some good evidence, particularly from Chile, on the effectiveness of the inactivated vaccine Coronavac, which has been manufactured by Sinovac. That was a very large real-world effectiveness study, which provided really quite reassuring data that after 2 doses, and it was clearly important to get the second dose, they saw very good protection in that population, in spite of the fact that they were continuing to have a lot of problems with the virus. So I think that proves the principle that we can expect these vaccines to be effective in principle. I guess we'll have to wait until this vaccine is actually, assuming it gets to licensure, is implemented before we can be confident about its clinical effectiveness.
Great, thanks. And there's another question from the audience about how quickly can the Valneva vaccine adapt to the variants?
Well, I think, at least in theory, the other platforms, adenoviral vectors and mRNA, given that they simply need the sequence of the spike protein in order to reformulate themselves, may be quicker. Nevertheless, the technology for developing a whole virion vaccine is very well established, straightforward viral culture. And so in principle, there's no reason why variant vaccines can't be produced. And indeed, that is ongoing at the moment within this program. I think the problem that confronts everybody, though, at the moment with regard to variant vaccines is not so much the speed with which they can produce them, but the choice as to which variant they should choose. Everyone shot off in the direction of what's now called the beta variant, the 351 that emerged in South Africa. But now everyone's looking in the opposite direction at the Delta variant, which emerged in India, which is rapidly rising in many parts of the world. And antigenically speaking, the 351 variants are kind of in the wrong direction from the original Wuhan and then the Alpha variant. So I think the real challenge for variant vaccines going forward is whether we can get the right ones and whether we can use multivalent variant vaccines to cover our options.
Fantastic. Well, thank you very much. And I think it's now the time to broaden our spectrum from the coronaviruses to other viruses of public health concern, right, Thomas? And you have some input to what viruses should we look at and what are the good options ahead on how to control some of these.
Yeah, thanks, Thea. Well, I've been asked to present on one particular virus. I just need to share my screen, and yeah, you should see the presentation now, I hope.
Thank you. Yes.
Okay, so I've been asked to talk about Chikungunya, which is not a virus that's so well known, but is actually an issue for people in affected countries, much more so, but also for travelers. So we've been concerned with the diagnosis for a few years now. And there is some development that's very interesting. So it's actually a good time to present on this virus, because we have a vaccine candidate that seems to be capable of changing the whole situation. Conflicts of interest. I have been doing studies and receiving monies from most vaccine producers that are involved in travel vaccines and other fields. And this, of course, is sponsored by Valneva, this presentation. So since I'm a clinician, let's start with the case report very briefly. It's happened in 2013, and actually I've seen the same patients just a few weeks ago. That's why I'm presenting. And at that time, 54-year-old man presented in our outpatient with ongoing pain in the joints, especially hands and wrists, just didn't go away. He was incapacitated really to work, couldn't work. He lived in the Dominican Republic as a journalist for the German press there. Couldn't type for the pain, so he couldn't work. And we established the diagnosis of Chikungunya by doing antibody tests. It was fairly straightforward because they had an outbreak at that time, so it wasn't so difficult. And I told him that the symptoms will disappear after a while. I gave him symptomatic treatment, felt that was an easy job, and basically forgot the whole thing. But he came again. Every 2 years, he's popping up, and latest was just briefly, and the symptoms did not go away. And by now, we know that that happens quite a lot, really. At the time, 2013, it wasn't so clear, the picture. Patient is still not able to work. So he had to quit his job, sell his house, and developed a depression, and is in a very dire financial situation. It's not a happy outcome, really, and there's very little we can do for him. Chikungunya is a bit of a weird name for a disease, really. It's coming from an East African language, the Makonde. It's a word for to be contorted, walk contorted, for the pain the virus is causing, really. Which can be different joints, really, not just the hand and wrist. It's been known to the medical world since '52, and it's been first described in an outbreak in Tanzania, and basically ignored since then, because it caused outbreaks, yes, but it wasn't really a large problem internationally. And it's an alpha virus, RNA virus, that's developed into 3 different strains now. By now, it's a West African, the original East African, and an Asian strain. And it's spread by mosquitoes, particular Aedes mosquitoes, Aedes aegypti and Aedes albopictus. And that's, of course, a clever thing to do for a virus, because these mosquitoes are spreading worldwide very effectively, and they're very effective transmitters of disease, really, since they are following humans and breed in urban settings, really, in cities, and they are active during daytime, but also at night hours sometimes. So it's a good choice if you want to become transmitted to humans, really. For 5 decades, basically nothing much happened, and then suddenly in 2006 the things changed. A mutated strain caused an outbreak, first in Réunion, in the French island in the Indian Ocean, and then started to move around, spread around the Indian Ocean to Southeast Asia, through Africa, and in 2013 jumped to the Caribbean, and then spread in Latin America and some parts of the southern USA as well. So it's been spreading very fast over a huge distance, really, because of course the vector was already there. Mosquitoes had spread before, and so the field was basically prepared for the virus. This is the first outbreak we had in Europe, in Italy, in 2007. Pretty early, actually. After the spread started, there was an index case here. There was an Indian traveler coming from India to northern Italy, and he was viremic but not symptomatic at the time. And he became bitten by mosquitoes in the area in the north of Italy, in the paddies there. And then suddenly cases started to pop up, and quite an outbreak developed. And that's the first 176 cases. In the end, it was more than 300. One person died, and the whole thing was just stopped because autumn started and mosquitoes stopped biting, really. And this is the area where chikungunya is occurring right now. So looking at the time frame from 2006 to now, it's been a very effective spread, really, in very short time. These are the reported cases in the US, and a huge increase in 2014, with a decrease later again. And these are German reports, which are similar with the same increase here in 2014, which was an outbreak here in many countries. It's not as frequently reported as dengue fever, probably because many cases are fairly mild, are not really detected, but it is there, and it is being imported into Europe quite regularly. Symptoms are or can be divided into an acute and a chronic phase, really, and we see very frequently pain in the joints, and basically all patients or most patients that become symptomatic. And then we have also other symptoms like fever, headache, muscle pain, conjunctivitis, nausea, vomiting, and others. In the acute phase, and that's most patients, we have a viremia for a short time, and then the symptoms resolve. And then, of course, in those who develop a chronic case, symptoms reappear. We have symptoms that are very similar to rheumatoid arthritis. There's obviously an immune effect here going on, and we're not quite sure what, really. And they can last for years or even forever, as it seems. And that's the problem for those patients, of course, when they are incapacitated and can't work anymore. There are some clinical pictures where typical, this is not by patient, but it looks very much the same, like this, with the hands that are it's just incapable to use, really. Here is the percentage of patients that have persistent rheumatologic symptoms, and as studies grow, we see that it's actually quite frequent. Réunion was a bit of an exception with over 90% ongoing symptoms, really, in this study, but it's not the whole population, it's just a subset, obviously. But it goes through basically all studies that we see in a large percentage ongoing symptoms in those patients, and there's very little we can do for them, really. In a small group, fortunately, we have also other manifestations, in particular neurological symptoms, which can cause severe complications like encephalitis, encephalopathy, Guillain-Barré syndrome, and myelitis. And these complications can also lead to sequelae, and they can be lethal. Paper published from Brazil on chikungunya deaths showed here split up by age groups that not many, but some patients die. And of course, older people tend to die more easily, which is the story of life. Basically, that's true for most diseases, but it's very much so true for chikungunya too. So if the age of patients increases, of course, then mortality increases too. Oops, I've been too fast, going too fast really here. There's no specific treatment really for the virus, so there's only symptomatic treatment. Up to now, there's also no vaccine to prevent infection, so the only thing we can tell people is not to become bitten by mosquitoes, which is a bit of a frustrating counseling really, because obviously it's very hard to avoid, in particular in mosquitoes that are breeding in urban settings, potentially close to where people live, and are biting at daytime when people are moving around. It's very hard to avoid those mosquito bites. If people have symptoms, we give painkillers, we give anti-inflammatory drugs, physical occupational therapy, and they need support, psychological support. And of course, it didn't help at the beginning when we told them things will go away, don't worry, and things didn't go away. So now we know that symptoms can persist and can counsel a bit more to the point, but there's very little we can do. There are more viruses transmitted by Aedes, and the symptoms may be very similar, in particular dengue, which is way more frequent even than chikungunya, and it's been longer around, and the new kid on the block in the last year's Zika virus. So all of these can cause fever and a rash. Joint pain is more pronounced in chikungunya, really. So that sort of sticks out really. And then in terms of severe complications, chikungunya is not as dangerous as dengue fever, but still people can die. And of course, we have many chronic manifestations. So a vaccine is needed. Unfortunately, there seems to be some development on the horizon, with a vaccine being developed by Valneva, which is a live attenuated vaccine strain, really, a single shot vaccine that's being applied and then appears to give fairly good protection against wild strains as well. There's a point mutation that caused the attenuation here on this gene. And that's been in a phase 1 study, tested in different doses to 120 volunteers, adults 18 to 45 in the US at 2 sites. And then they used a challenge with the same vaccine in the highest dose group and gave them again the vaccine just to see whether there would be another viremia or whether there would be complete protection. It's not of course a wild virus challenge, but the best you can do probably in a study setting outside an outbreak situation. And that's the publication that provided the outcome, published last year of that study. These are the effects with the different doses here, and all subjects responded nicely, really, and had a 100% seroconversion. And these are the side effects, which were giving that is a live vaccine, and of course there can be fever in live attenuated vaccines. We see that in yellow fever, for example. There can be flu-like symptoms and so on. Given that symptoms were comparably mild, very few reports actually in all groups, in all dosings. And on the challenge group, this is the right lower here, there was a bit of an increase of nausea, in a judged severe in some instances, but that was about it. So overall, it was very well received, the vaccine, and in that study, 120 volunteers, also pretty effective. This is the viremia after the challenge in all groups, increased in the high dose group, obviously, and this is the viremia in the after challenge, which was 0. There was nothing detected, judging on the good protection of the immune response.
Sorry, we just have 2 minutes left, just making a note of it if we...
Okay, I'm just finished. So summing up, it seems to be a highly immunogenic vaccine, as phase 3 trials running, and it seems to be very safe. And just 2 days ago, the US approved the vaccine for breakthrough designation, so we can expect results soon from phase 3. Thank you for your attention.
Thank you very much, Thomas. We just have time for one question. So in the audience, there's a question: how do you believe current pandemic is affecting the perception of risk for other travelers' disease?
I think the current pandemic is pushing away risk perception for other diseases. So we need to work on that to straighten the picture a little bit. Of course, the current pandemic is important, but it's not the only disease we have, really. And Chikungunya is just one example. We have been ignoring, in particular, diseases that are a problem for poorer countries in the West, because we are not affected that much. It's dengue fever, chikungunya, and others. And we need to straighten the picture again a little bit and concentrate on those diseases as well. And in fact, in some instances, those have even benefited those diseases, like malaria has been on the increase in some countries because control programs have been stopped and others.
Well, thank you very much for a nice presentation and also for answering the first question. And unfortunately, time is out, but it has been very interesting to chair this session. Thank you very much to both presenters and to my co-chair. And hope that this meeting will allow for us to meet in some of the other sessions, and we can actually still continue some of the questions.
Thank you, Thea, for supporting us on this session. And also from my side, thanks Thomas and thanks Adam, and thanks to all our participants. Goodbye for today.